Hey guys, I'm Aisha and it's my first video on YouTube where I'm gonna talk about something educational, and it is about medical science, a chapter on biochemistry. Like I'm gonna talk about a topic of biochemistry, okay? And from the first slide you can guess what's the topic. It's about nucleic acid, okay? So when you think about nucleic acid, what is the first thing that comes in your mind? For me, when I think about nucleic acid, I think that what is the nucleic acid we have in our body? So, the nucleic acid we have in our body is DNA and RNA, right? I think you already know that. And from the abbreviations, you can already guess or already know that it's obviously a nucleic acid because DNA abbreviates as deoxyribonucleic acid, while RNA abbreviates as ribonucleic acid, right? So there is not much anything to describe in this slide. So I'll move forward to the next slide, okay? So before we move to the chemical structure, we'll talk about the physical thing about DNA. Like DNA has two strands which lie antiparallel to each other, and these two strands intertwine with each other to form a staircase. You already know the structure of DNA, I think, right? Still, I'll draw it like this, right? You can see the intertwine strands like the fashion of the spiral staircase. And about the antiparallel thing, I will say that imagine this is a strand of DNA. This is the 5' end and this is the 3' end. And remember that DNA always goes to the direction of 5' to 3'. So here is the another strand of this DNA, and this goes the same way as 5' to 3', but they are antiparallel cause these go this way, okay? Like you can see they are going to the opposite directions, and that's why they are antiparallel, okay? And in case of RNA has just one strand, okay? So that is the physical thing you need to know. That's the basic thing of DNA and RNA. Okay, so I'll move to the next slide. Uh, as both DNA and RNA is a nucleic acid, so we need to know what is a nucleic acid. So, in chemically speaking, a nucleic acid is a polymer of nucleotide. And what is a nucleotide? You might say that it's just a monomer of nucleic acid. Well, you are right, but that's not the answer a teacher would like to hear. Okay, so what would be the correct answer of nucleotide? What is nucleotide? So nucleotide is formed by a base, and specifically speaking it is a nitrogen base. We'll talk about nitrogen base in next slide. Okay, so nucleic acid is formed by base, sugar and phosphate. Here we can see three phosphates, okay? So it is like triphosphate, and it's a nucleotide. So nucleotide is formed of base, sugar and phosphate. We already know there are two types of sugar, the ribose and deoxyribose. DNA has the deoxyribose sugar where RNA has the ribose sugar. Okay, and what is nucleoside? Nucleoside is composed of a base and a sugar. It doesn't have any phosphate. So we can also say that a nucleotide is a phosphorylated nucleoside, because when we add phosphate to a nucleoside it becomes nucleotide. So I can say that a phosphate with the nucleotide, oh sorry, phosphate with a nucleoside, it will be a nucleotide. I think you have got that. Okay, so we'll move to the next slide. The N bases, the nitrogen bases. Okay, so there are two types of nitrogen bases, the purines and pyrimidines. Purines have two rings where pyrimidines have one ring, and you can already see that in picture. It have two rings where it has one rings. So if you want to know more about the biochemistry, you should at least know the structures. I'm not saying that you have to memorize them, but I will just say that remember them. It will help in future. Okay, so I will say about some easy techniques to remember the structures. Like purines has two bases. Like there are two kinds of purines, the adenine and guanine, and there are three pyrimidines: cytosine, thymine, and uracil. And our DNA has the adenine, guanine, cytosine, and thymine, and our RNA has adenine, guanine, cytosine, and uracil, and not the thymine. So how will you differentiate them? You can differentiate purines and pyrimidines by the structure, that the purines have two rings and pyrimidines have one rings. But how will you differentiate adenine and guanine? You can just differentiate by seeing the amino group. You can see amino group in 12 o'clock position of adenine, but it doesn't have any amino group in guanine in 12 o'clock position. It has a amino group in 7 o'clock position, but that is not in 12 o'clock position. And amino group in 12 o'clock position is your key to differentiate. If it has amino group in 12 o'clock position, it is adenine. If it doesn't have any amino group in 12 o'clock position, it is guanine. Same goes for pyrimidines. Okay, when you think of pyrimidines, you have to think about the one ring at first. Then there comes if it has an amino group in 12 o'clock position, then it is cytosine. If it doesn't have any amino group in 12 o'clock position, then it will be uracil or thymine. Now how will you differentiate the uracil and thymine? You can see that the uracil doesn't have any methyl group, but the thymine has a methyl group, and that is your key point to differentiate the uracil and thymine. When you add a methyl group in uracil, it becomes thymine. Interesting, right? Okay, so I think I have already said everything you need to know about the nitrogen bases. So I'll move on to the next slide. So here comes the sugar. I have already said about the sugar that the DNA has the deoxyribose sugar, where the RNA has the ribose sugar. Why we say deoxyribose? Because it has one less oxygen, where it has a, an extra oxygen in ribose in 2 number carbon, where that oxygen is missing in deoxyribose in 2 number carbon. And that is the key point. Differentiate the ribose and deoxyribose. And that is the main difference between DNA and RNA, of course, because DNA is composed of the nitrogen base, deoxyribose sugar, and phosphate, where RNA is formed by nitrogen base, ribose sugar, and phosphate. There is the difference in nitrogen base too, and we have already talked about it in the previous slide. So I'll move to the next slide then. Okay, so there comes the phosphodiester bond, and specifically speaking, that is 3,5 phosphodiester bond. So why we say 3,5? It's not a detail you need to remember, it's just a basic you can remember. Okay, so this is a DNA which going from 5 prime to 3 prime. This is the 5 prime end and this is the 3 prime end. In this sugar, this is the third carbon, and the 3,5 phosphodiester bond is with this 3 carbon, and this is fifth carbon. Okay, that's why mainly this bond is called phosphodiester bond. Now you may think why I'm talking about a phosphodiester bond suddenly. Okay, so we said that nucleic acids are a polymer of nucleotide, and nucleotide is monomer of nucleic acid. So nucleotides, if we say NT for short, the nucleotides need to be connected by a bond together, right? And then it will be a nucleic acid, and that is the phosphodiester bond. You just need to know the name, nothing else is much important for the exams, just the name, the 3,5 phosphodiester bond. Okay, so I'll move to the next slide. So here comes little complex thing. Mainly the figure is complex, but if you get it once, and if you get the previous structure of nitrogen bases, then you will get it too. Okay, so at first I will say about the nucleoside and nucleotide to just clear the concept little more. Okay, so here we have the base, the adenine, and when you add sugar, when you add ribose sugar to the adenine, it becomes adenosine, and when you add deoxyribose sugar with adenine, it becomes deoxyadenosine. Same goes for guanine. With addition of ribose sugar, it becomes guanosine, and with addition of deoxyribose sugar, it becomes deoxyguanosine. And same goes for others. But the thymine, as it is only in DNA, it can add with only the deoxyribose sugar. So the thymine is deoxythymidine. The uracil has uracil with the addition of ribose sugar becomes uridine, cytosine becomes cytidine, and so on. Okay, and When we talk about nucleotides, it is about the phosphates. Nucleoside is phosphorylated, becomes nucleotide, right? That's what we talked in the second slide or third slide, maybe. Okay, so you can see that adenosine monophosphate, but one phosphate is added to the adenosine and becomes adenosine monophosphate. 2-phosphate is added to the adenosine and becomes adenosine diphosphate, and 3-phosphate is added to the adenosine, it becomes adenosine triphosphate, and all these things goes the same way. Now we will talk about the structure. So let's focus on the first figure. Okay, so at first you can see that this picture has two rings. So as it has two rings, it's a purine, and it has amino group in 12 o'clock position. It is adenosine, like adenine. This is adenine, and with the addition of ribose sugar, this is adenosine. And when you come to the second picture, it has one ring, so it is pyrimidine. It has the methyl group, so it is thymine, and it doesn't have oxygen in the second carbon, so it is deoxyribose. So the full thing is deoxythymidine. You can get it right. Okay, same goes for the next picture. This is the purine because it has two rings. It doesn't have amino group in 12 o'clock position, so it is not adenosine, it is not adenine, so it is guanine. Okay, and there is no oxygen in the second carbon, so it is deoxyribose, and there is a phosphate too. So what is it? It's guanine, deoxyribose sugar, and phosphate. So deoxyguanosine monophosphate, you can see it. Then there comes the last one of this slide. Here is a one ring base, and it doesn't have the amino group, and it also doesn't have the methyl group. So what is it? It is uracil, and it has the oxygen into number carbon, so it is ribose. So the full thing is uridine monophosphate. I think you got it. So let's move on to the last slide. This slide is one of my favorite slide, and it has almost everything you need to know about DNA. Okay, so at first I will tell you to think, is it a DNA strand, or it is a DNA RNA strand? You can answer it if you understood what I talked all this time. Okay, so I will zoom it for you a little so that you can think. Okay, and you can see that these two strands goes to the 5 prime to 3 prime direction, right? This goes 5 prime to 3 prime this way, and this 5 prime to 3 prime goes this way, so they're antiparallel, right? Okay, now comes the answer of the question. Is it a DNA strand or is it a DNA RNA strand? So let's focus, okay, on the picture. So you can see that there is this nitrogen base. Sorry for that text. uh, okay, never mind that. So I was saying that focus on that nitrogen base. It has a methyl group, so it is thymine, and DNA doesn't have thymine. Sorry, the DNA has thymine and RNA doesn't have thymine. So this is a DNA strand. Same goes for the second strand. It has the thymine here, so it is also a DNA strand. This is the easiest way to think. Also, you can check the sugar if it is deoxyribose or it is the ribose. Check the second carbon. Here it doesn't have the oxygen, so it is deoxyribose. Here check the second carbon. It also doesn't have oxygen, so both of these strands are DNA and DNA, so it is a DNA strand. And also here in between these strands there is a bond here. What is this bond? I think you can guess that this is the hydrogen bond, the triple bond existent between cytosine and guanine, and a double bond existent between adenine and thymine. Here we will focus on the last thing, the last bond, the phosphodiester bond in between the 3 number carbon and in the 5 number carbon of the sugar. There is the phosphate, and it is creating the phosphodiester bond. I think I have said almost everything you needed to know in this lecture of nucleic acid, like the compositions of nucleic acid. So I'll conclude this lecture here. So thank you for watching. See ya.